Moving on, it is my pleasure to introduce our next speaker, Dr. Vaibhav Dixit, Assistant Professor from NIPER Guwahati, who will speak on the topic Predictions of Hard and Soft Spots using Molsoft, Drug Metabolism, and Covalent Binding Application. His research focus areas are ML guided drug design and drug metabolism, prediction, and applications of medchem principles in anti cancer drug discovery. Let us welcome Dr. Weber with a round of applause.
I should take this opportunity to thank Surojit and Andrew and the Molsoft team for inviting me and giving me a chance to present our work. As he showed that we have just in the last 1.5 years we have started using this tool. I was already aware of it for some time, but we were already having some other tools in our place. So only when my student told me that we should work on covalent inhibitors, said I immediately agreed, and we started working on that. And yes, so we are here. So thank you once again. So without wasting much time, I will directly jump into it. I have been working in the drug metabolism prediction area for some time, and we have recently got some success, and therefore I tried to bring those things together and how we can use Molsoft and other tools that are available for predicting hard and soft spots, and it has also applications for covalent binding prediction. And there is a disclaimer, so which is standalone. So in one of the Ministry of Electronics and Information Technology sponsored project, we were working on anti-cancer drug design, and we were more. So as you know that in the academic environment, it takes some time to get meaningful results. So I thought that we should first review the literature and then understand what are the problems of the existing drugs. That way the students will get opportunity to learn, and we will also have some good insights we can develop into existing problems. So as you know, I will skip the introduction about cancer and breast cancer that most of the audience is aware about. And these are the four drugs that are currently available in the market and very popular. But they have, because of the last 10 and plus years of clinical usage, now the toxicity and resistance have started to emerge. And we have there have been reports of hematotoxicity and hepatotoxicity and also cardiotoxicity, especially with olaparib and other molecules. This is not to say that the drugs are not useful. They are useful. The company knows what they are doing. We are, from an academic perspective and from a scientific perspective, we are focusing on why and in somehow if these become toxic, why they would become toxic. So and always whenever there is metabolism involved, there is always a chance of reactive metabolites generating, and that precipitating a lot of toxic responses in some patients which are more susceptible to those things. So so when we did literature search, we found that there are three metabolites which are predominantly reported in many of the rat and mouse model and also in humans, and most of them are through amide hydrolysis, hydroxylation at the piperazine ring and the fluorophenyl rings. So we said that we will investigate this using a combination of accessibility and reactivity approach. So for reactivity, we used quantum chemical calculations, and we have performed these calculations on our own in-house HPC, which we have coined it name called a Param embryo, because our director is very fond of this Param series of supercomputers, and we have some long-standing collaborations with CDAC. As soon as I realized that, then we started calling it Param embryo, and you know how it goes. And so this is the overall summary. In the literature, M11 is known to be the major metabolite, but it is not toxic. So there the question is, can M12 and M15 be toxic? Mechanisms involved in their metabolism formation, they are were properly not known with some details, and the question still remained that which of these is going to be reactive metabolite and which of these might be associated with toxicity, and can this reactivity and accessibility approach help us answer these questions. So moving forward, we did quantum chemical calculations on this metabolic pathway, So in the first step, you see that the compound 1 as the cytochrome, I have not gone into the details of the catalytic cycle of the cytochrome. If you have any questions, I will be more than happy to answer that from the audience. So as in the first step, what happens is that once the molecule is inside the active site and compound 1 reactive center is ready to metabolize it, you will have the transition state where the proton will get abstracted by the oxygen of the heme center, and then the radical will form. The radical will rebound back to give a rebound M11 complex. This simply dissociates and gives us the hydroxylated metabolite. And similarly, a similar sort of oxidation happens for the aromatic ring, which is at the bottom part of the screen. Here. And here also you see some, in this case, the oxygen attacks on the aromatic ring followed by 2 proton rearrangements, and that complex is then thrown away and you get the hydroxylated metabolite at the aromatic ring. So this is the potential energy diagram. It is much crowded, but the overall message is that the transition state for the formation of M11 is really high, around 24 kilocalories per mole, and at room temperature or even at body temperature, anything which has a barrier of 20 kilocalorie or less is going to be feasible, thumb rule kind of. So when this is 24, it is not possible. So according to reactivity, we can rule out that formation of M15 is going to be really slow process and it won't happen. For M6, it is within the cutoff of 18 or 20 kilocalories. So this is a likely process, and the formation of M11, which is highly feasible, it is hardly any barrier, almost barrierless. So that explains the formation of M11 as a major compound, and lower amounts of M15 can also be explained based on reactivity calculations and parameter estimation. But why M6 is a major metabolite despite having favorable energetics? So this is not clear. Reactivity is giving half of the answer, but still some conundrum still remains there. For that, we have used accessibility and we did docking. For this work, initially we did docking in some other software, but then we repeated these docking calculations with Molsoft tools and predicted the ICM Pro interaction energies. And here is a summary that these bold values you see in the table, those are the lower ones, and these are the top 3 poses we are considering for comparison. In the published paper, we have considered up to 10 poses and made an extensive explanation based on what seems logical. So so distance stands for accessibility, how close the metabolic site in the molecule is able to access the reactive center of the cytochrome, and this reactivity is accounted by the enthalpy of the reaction, quantum chemically calculated reactivity. So here we say that for M6 distance is not favorable. So even if the energetics is favorable, the molecule is not able to enter into the active site in that orientation. So even if the energy will not prevent its formation, it is not able to gain access to the reactive center of the enzyme. So that kills. For M11, both criteria are yes, that means it is able to gain access into the active site, and the energetics also they are favorable. So both is happening. But for M15, the distance is neither distance is favorable nor the energetics is favorable. So reactivity and accessibility together allows us to explain why certain metabolites are major and why certain are minor. Then we did not stop there. We went and did using those docked poses, we did a MD simulation, because the distances, you might say that might be some not the whether that distance is really stable over a period of time during the MD simulation. So we did that, and we are able to consistently see that the reactive center, which gives the major product, that is coming closer and remains closer, and the metabolite which is formed in minor quantities, it comes within the reacting distance only few number of times. So it's not like it never comes closer. It does come closer, but very few number of times during the MD simulation. That would suggest that if a patient has a mutation in the active site, that now suddenly prefers that pose, now it will have energetics and also access in the active site. So that patient might show idiosyncratic reaction because they are able to form higher amounts of the reactive metabolite. So there is another way of estimating reactivity of the molecules using quantum chemical calculations. We did global and local electrophilicity calculations, and we in-house we build scripts that allow us to generate these nice figures. And here at the below the structure, you are able to see the global electrophilicity value, and the circle, blue circle, they represent the electrophilic centers. The larger the circle, the more the electrophilic the atom in the molecule. But that you have to see along with the electrophilicity value of the molecule. So this is this is olaparib. Its electrophilicity is really small, 1.96 compared to some of the reactive ones. This is M12, which is the most reactive metabolite. Overall electrophilicity is very high compared to the drug itself. So when the drug gets metabolized to this molecule, it is really reactive, and it will start reacting with macromolecules and nucleophilic sites in the body and will cause toxicity, may cause toxicity. So here, and then there is a overall final answer. This is a student. She is even now 5 minutes ago I was interacting with her and trying to finalize some of the data that is in the next slides. So she is one of the brilliant students who we recently managed to publish this work in Chemical Research in Toxicology. So now moving this, using similar ideas and connected ideas in another project, we are trying to design covalent inhibitors. And this work is going in background in my mind, but only when Surojit called me that you have to come and present, I said that this only one story is not enough, I have to build something. And since that, even the work is still going on, she is doing something. And but the current status I will share here. So as we all know that covalent inhibitors are very interesting. They have gained interest in the last 5-10 years. And as people have understood what makes them toxic, what makes them selective, and people are confidently able to convert them into drugs. So everybody is interested in designing covalent inhibitors. And the basic principle revolves around this. First, there is a reversible binding, and once the molecule gets access into the site, then the covalent bond forms gradually. So there are two processes are happening that one needs to account for to understand this in greater detail. So these are the well-known approved drugs and some warhead groups that one is usually interested in. So recently I looked into the literature and tried to develop my own understanding of how this one paper JMC cited at the bottom is very helpful, demystifying factors that influence covalent binding, where the editor have given these explanations. So the main difference between reversible and irreversible binding is the time dependent formation of the covalent bond. The kinetic measurements are required to make accurate predictions. And so covalent binding also involves a non-covalent binding component that is given by Ki. There are two Ki's here, one Ki with a small I and another Ki with a capital I. So you have to be careful about that. And there is another factor that comes into the equation is the residence time. The inverse of that is the K off, or the dissociation rate constant. So all of these can be rearranged into this nice equation, which this is the inactivation constant that you would measure for a covalent inhibitor. It is governed by these three factors which are in the equation. This Ki is the reversible binding for the covalent inhibitor, and this K inact is the rate at which the covalent reaction happens, and tau is the residence time, which is nothing but the inverse of the K off. So if you are able to predict these three accurately, you immediately get that. So this 10 days ago this idea struck me, and then since then I have been working on it, and we have only preliminary data that I will show. So here the best target to choose is EGFR, because there is a lot of data available on it, lot of drugs and lot of experimental data is easily available in the literature. So I immediately jumped onto that. So without going into the details that most of you understand, so these are some of the first and second generation and fourth generation work. So using Molsoft 3D QSAR method, we collected a data set of from Campbell, which contained 25,000 molecules, and then we filtered this into a single protein. So we got 528 data points, then data pre-processing, we removed duplicates, removed salts, then it reduced to 324 data points, and then we filtered the warhead group containing compounds from this. So only 64 molecules remained, which are purely reversible binders. They do not have covalent binding. So using that, we build a 3D QSAR model, and that gives me R square is equal to 0.83. So I might ask the audience, would you be happy with this model? Would you like to improve it further? I would assume that using AI ML tools, you will want to improve it further. Yeah, anybody disagree? What you would generally do if you generate a data set, reliable, high quality data set, and you get only 83%, 0.83 R square, would you be satisfied with it, or you want to increase it further? Huh? Increase it further would be the natural answer. So yeah, that's what the student came up with the solution, that let us build a QS, let us generate thousands of descriptors and try to use standard machine learning. And I said, yes, you can do that, but let us do something else. And I said, then what exactly to do, I could not tell her. So that day we stopped the discussion and I went to sleep. I could not sleep. I kept on thinking, and this thing came to my mind. And I'm now calling it unbiased R square, a novel concept to avoid overfitting. So ML model performance is based on R square when you are doing regression, right? So, and this R square value is calculated using that formula. So this, I would claim that this metric is biased towards experiment and expects a perfect prediction from the model. It's like a parent who is expecting that you have to come 100% in that exam and you have to be an IAS officer or whatever. But I am not required to be perfect, but you have to be perfect. So it's being biased, isn't it? So the real world data comes with error bars, and those should not be ignored. They should be considered, right? So IC50 values have built-in standard deviation, and around Campbell, if you do search of Campbell dataset, you will find that the standard deviation could be anywhere between 0.33 to 0.69 log units, maybe even larger. Depends on what is the target, what is the assay, who is doing the assay, how well it is validated, what confidence do the experimentalist have in it. So R square values, the traditional R square and the formula, it ignores this standard deviation. Thus, I propose a novel unbiased R square, UR squared. I am calling it. You are not cornered, you are squared. I don't know what it means. I am still thinking over it. Yes, I am also squared. So, this UR squared, I am just replacing the Y predicted in the numerator by something called Z, and the Z can be these things. If your Y predicted value is more than equal to the experiment plus standard deviation, then you set the value of Z is equal to Y predicted minus standard deviation. If the Y predicted is less than experiment minus standard deviation, then set the value of Z equal to Y predicted plus standard deviation. If the Y predicted is within the experimental standard deviation, you set Z is equal to Y experimental. And this might be confusing, that's why I built this figure. And the blue circles are predicted values. No, the blue circles are the experimental values, but they come with standard deviations. So if you look at each, this is the experimental value, and this this is the predicted value, and it is this is the standard deviation of the experiment. Okay, so my prediction should also be allowed that much deviation. My prediction has to be perfect. The prediction should be as good as the experiment, and as soon as your model is as good as the experiment, you are done. There is no need for modeling further, I would claim. So, and with this figure, I want to put that point in front of you, and I am proposing a new metric, and just calling it UR squared. So this, so don't use this predicted value in your equation. Instead, you use that value because it is within the standard deviation. Similarly, for this, don't use this predicted value. Instead, you use that value because it is still within the standard deviation. For these, then do not fall on the line, but nevertheless, why to use this value? Using this value would make more sense because it gives you higher UR squared. So your model is not 88% accurate, it is actually 96% accurate, 98% accurate already. Yes, it is already 99% accurate if I can use percent for R squared. Okay, so there is no need to so UR squared will consider any prediction within standard deviation as perfect prediction, as accurate. This leads to higher UR squared values, and the model already performs well within the experimental standard deviation. There is no need for further improvement. And then I immediately, before claiming that it is my own novel invention, I searched literature and I found one paper in 2012 where Kramer proposed a maximum Pearson R square, which is very similar, but it's not identical. So I still have my own novelty in it. But it didn't consider limitations imposed by the experimental errors on individual analysis ML models, and considers only the database averages. Whereas UR squared allows you to do case by case analysis, and it indicates overfitting. So if your R square is by somehow more than UR squared, that means your model is already overfit. It's not good. It's somehow bringing you learning stuff that it should not be learning because it is hallucinating in its own world. So better not do that. So they often consider can be improved, but actual improvement, and what is the actual improvement that is required? It is not 1 minus R squared, but it is 1 minus UR squared. That is the one that is actually possible. So some of the points might be getting remitted. So EGFR data set, the Molsoft tools and the 3D QSAR is already giving a model that is already within the experimental error. So why to improve it further? First, you go back to the lab and improve the experiment. It's not easy. It's easy for me to say from here. So this is how I would like to summarize this part, that if you calculate the R square traditionally, you will expect that the perceived improvement that are possible are remaining 17%, and with an experimental error of 0.55 log units, the UR squared becomes 0.987, and the possible improvement is only 0.1%. So there is no need for ML or deep learning modeling here, according to me, although no experienced people are sitting here, more 20 years more experience than me, so we can debate on that. So now back to so using this QSAR model, I have predicted the Ki with a small I, but now I want to predict the rate constant for the covalent binding. So for that we did we are using Meyer's equation, which predicts you take electrophilicity and nucleophilicity value from the quantum chemical calculation and multiply it with some cofactor, and then you get the rate prediction for the covalent reaction that is happening between two. Here also then I got a model that was already 86% accurate, but again here somebody will say that no, this is not good because you need 95% accuracy. Then I said that let us apply the UR squared here, and it is actually already 1 here. So if something has to be improved here, it is the experimental measurement that has to be improved here. Why to unnecessarily overtrain our models when they are already better? So then combining all these three information in one, we are able to make some predictions, although this is the predicted value Ki. It is not a perfect match, but it is, I believe, this is still work in progress, because for these compounds the residence time is not known. So I have assumed a residence time of, that's, I was just typing these 10,000 when I was a couple of minutes ago. So this work is still in progress, that from the experimental data, can you build a model which predicts accurately Ki value? Can you build a model which accurately predicts the K inact? And then somehow you would also need a model that predicts the residence time. Combining these three will give you the equilibrium rate constant for inactivation by a covalent inhibitor. So what are the future conclusions that and future direction? That reactivity and accessibility is useful. Reactivity allows us to identify reactive centers. Accessibility helps us to predict accurately sites which have access to the cofactors or residues inside the protein. A combination of these two factors is what is required to make accurate predictions. Don't corner regression models, you use R squared to avoid overfitting. And so these concepts of accessibility and reactivity can be combined and used for design of novel covalent inhibitors. We are actively working in this area, and we have some collaborations with pharma industries and CROs, which I have not mentioned here because it is yet to fully finalize. We are still working on that. And here is a glimpse of what other work is happening in the lab. In these ministry sponsored projects, we have developed machine learning models to make PARP1. I have to apply UR squared to these also. That part is remaining. So using these models, we have built web servers and we copyrighted the software. And we in the drug metabolism prediction area, we are also, although there are all these software companies that they sell you ADME softwares and they tell that metabolism is predicted, but they are already only doing substrate or inhibitor prediction. They are not telling exactly where, at what site the molecule will get predicted. Some tools are available there, but they have their own limitations. And we first identified the niche area, that site of sulfonation, for which there were hardly one model was available, and we were already building that. So we completed this. There also we have used the reactivity and accessibility approach to build highly accurate models, and we have developed our own algorithm and bundled it into a user-friendly software, for which we have also done a copyright, and we have also collaborated with the National Research Development Corporation, NRDC. They allow government organizations to out-license their tools and techniques. So this SOS predictor software that we have developed, it is available through NRDC to third party. So anyone interested in phase 2 drug metabolism prediction, am I allowed to sell my own software? I don't know. So if you are interested in drug metabolism prediction, site and non-sites, and we can collaborate, we can think about something, maybe if these tools can become part of Molsoft, not so soon. We'll work on that. So once again I thank Advent Informatics team, Surojit and Pritha, and the Molsoft team. My director, he has always been very supportive, and whatever I have asked him so far, he has given. So yeah, that's all good happening. And we also acknowledge the Ministry and Department of Pharmaceuticals and all the students and their work. I am guiding that work and mostly getting involved whenever possible, and I am open to questions and queries. Thank you so much once again.
Thank you, Dr. Vaibhav, for that wonderful presentation.